Everyone, and thank you for joining us today for our webinar titled Gait Analysis in Laboratory Animals, Studying Coordinated Movement and Associated Disorders. My name is Martin Hess from Inside Scientific, and I will be your host for today's event. We are happy to share that we have over 200 scientists from around the world who have registered to participate in this webinar today, and I would like to personally extend a warm welcome and thank you to all of you who have taken the time out of your busy schedules to be with us. Our session today is sponsored by Mouse Specifics and will provide our audience with an overview of important parameters for studying gait in laboratory animals and specific examples of how gait is used as a quantifiable measure in research laboratories focused on mouse models of amyotrophic lateral sclerosis, ALS, and Parkinson's disease.
Well, thank you everybody for signing up for the webinar and sticking with us for the first few minutes this morning. I'm very passionate about this field. I'm a researcher myself with aging parents and 4 children, so I'm very interested in maintaining my ability to walk and run. So I'm actively engaged in the study of animal models of human disease to measure strength, balance, and coordination. What is gait analysis? Well, it's the study in a quantitative way of the manner by which we walk and run. And people have been studying gait in mammals, human beings, and racehorses primarily for well over 100 years. And when is it disturbed? Well, I imagine we all know a relative with Parkinson's disease or brain tumor or multiple sclerosis, or we ourselves might have an injured knee or a nerve injury or know somebody with a stroke. There are countless examples of when our motor capacity is disturbed. And today, each one of our speakers will focus on amyotrophic lateral sclerosis. Dr. Carol Milligan will present more on that, and Dr. Charlie Meshul will speak on Parkinson's disease. And why gait analysis? Why am I bringing this up with regards to the mouse models? Well, people have been studying tens of millions of rodents now for decades. And there are a lot of tests out there for looking at motor function in lab animals. For me, the video on the bottom right with the log rollers is the closest I can think of how rotarod might translate to the human condition. But patients in wheelchairs and subjects on crutches, what they really want to do is walk. So about 8 or 10 years ago, we decided that there needed to be some good instrumentation to quantify exactly how it is that these lab animals are walking so that the murine data can translate to the clinics faster. And why treadmill gait analysis? There's other ways of looking at gait in lab animals. Well, for starters, rodents rarely follow our instructions. I'm sure there's people in the audience that have found it frustrating to have the animals not necessarily do what we hope that they'll do. The walking speed significantly impacts posture and kinematics. The treadmill allows us to get data from animals walking at comparable speeds. So we have higher repeatable data with lower standard error. And we can explore challenging speeds, just like we do when we're at the physician's office and maybe we're asked to get on a treadmill because the challenging condition may reveal something subtle or something early. So what does gait analysis measure? Well, certainly in human gait analysis labs, of which there are many, the gait analysis is used to quantify the stride length of the subject. How far apart are the feet? Are the toes pointing inwards or outwards? Is the swing time exaggerated? Is the subject steady on his feet or her feet as they walk. So we developed DigiGait for looking at kinematics, but an elegant method whereby with one camera we can view the ventral side of the subjects as they walk on a transparent treadmill belt. So by analogy, imagine any one of you at the health club or the gym walking on a treadmill, a camera looking up at the underside of your sneakers, taking high-speed photographs of where your sneakers are with respect to the treadmill belt over time. And so what that camera is going to see and map is the area of your sneaker with respect to the treadmill belt over time. But in the case of the lab animals, we're going to generate 4 such gait signals. And those signals describe how long a foot is in contact with the treadmill belt, how long a limb is in the air, the complete stride time. And looking at contact time, we can further delineate a slowing down phase of the animal. So if you're all sitting down, put your heel on the ground, and this is the loading portion of your limb, and this is the unloading portion of your limb, getting ready and executing into the next stride. So the artificial intelligence algorithms of the DigiGait software determine the spatial and vector components of the paws relative to the treadmill belt, resulting in gait signals for each limb. So a picture of a mouse walking on the DigiGait treadmill, and again the camera's looking up. We can also image from the side, but most of the data comes from the ventral view. And the advantages are many, but they include the ability to calculate our metrics based on multiple strides. If we turn the treadmill belt to 0, we can look at voluntary walking, but it's much more interesting to explore a range of speeds, anywhere from 0 all the way to 100 centimeters per second. Most laboratory animals are in the range of 24 to 45 centimeters per second, but of course mice that have been bred for high speed endurance reach 99 centimeters per second. And we can look at horizontal walking, incline walking, and decline walking. So I mentioned gait analysis labs for humans where stride length, stride frequency, stance width, these kinds of things are used diagnostically in patients. Now we're able to generate these same things in the lab animals. Stride length, stance width, cadence, paw placement angle, swing time, step-to-step stability, braking duration, propulsion duration, gait symmetry, paw overlap, paw area, tau, rate of loading, sciatic functional index. Whatever the camera sees, and we're capturing at very high frame rates, the software will compute. So what kinds of things can we comment on or report or publish or describe? Well, what I show here is a very simple experiment where a guinea pig was studied at 2 different speeds, 18 centimeters per second and 36 centimeters per second. Well, the software reveals to us not only that differences in walking speeds significantly impact things such as stride angle, stride length, and paw placement angle, but also the coordination of the animal is changed at the increased speed. Here's the ventral view of a normal, healthy rat walking on the DigiGait. This animal, like most quadrupeds, follow an alternate stepping sequence. And the gait signals generated by the software describe to us in very qualitative way the differences between what the forelimbs are doing. So here are the gait signals for a left forelimb, right forelimb, left hind limb, right hind limb. Well, let's look at the slopes of the gait signal for the hind limb. The heel comes down very abruptly, the limb is loaded very quickly, and the push-off phase is more of a process. This slope is much more gradual than the upstroke in these hind limbs. The forelimbs, it's almost the opposite. The forelimbs come down more tentatively, and they release much more quickly. And these qualitative findings, which are reflected in the quantitative metrics, dovetail with what's known about rodents. They don't see very well. They hang out in the dark, and they use their forepaws for navigation and somatosensory feedback and proprioception, and the hind limbs are used more for the flight and fight response. And taking that animal, that healthy animal, and looking at the gait signals for the hind limbs and the forelimbs, taking an ensemble average of numerous strides and superimposing those data, we can see here in a normal healthy animal that there's excellent symmetry between what the left hind and the right hind are doing and what the left fore and the right fore are doing. And this becomes very useful when we're looking at models of unilateral injury. In this example that I show here, this is a nerve crush model where crushing of the right sciatic nerve has caused a collapse of the posture of the right hind paw. So if you see that in comparison to the left hind, you can see that the posture has changed. Well, that's pretty clear. But down here, DigiGait is able to show us how the kinematics are affected. Not only is the actual support area seen by the injured limb markedly reduced, but we also see, and this was a new finding, compensatory changes occurring in the forelimb as a consequence of the hind limb defect. Here's additional data from a mouse model of ALS and Parkinson's disease, I guess as a means for introducing my speakers. But the point I want to make here, if you look at, if you all hold up in front of yourselves 2 fingers and approximate a ½ centimeter, that's the kind of reduction of stride length somebody might expect in a mouse model of Parkinson's disease or a mouse model of ALS. In humans, for example, with Parkinson's disease, stride length reductions are on the order of 10%. Scaled to the mouse world, a 10% reduction in stride length is a ½ centimeter. So expecting an animal to walk voluntarily in a consistent manner enough steps to reliably teach us or show us a ½ centimeter reduction changes is challenging indeed. And so that's one of the great benefits of DigiGait, is to have all the animals walk the same speed for continuous steps so that we can see these magnitude changes. But also in the case of ALS, we're quantifying and seeing significant changes in hind paw placement angle, a good 4 or 5 weeks sooner than rotarod is evidencing any defect in the SOD1 G93A mouse model. And rather than concentrating on stride length in Parkinsonian models, DigiGait allows us or empowers us to look at changes in stride-to-stride variability. And so we can see here in this neurotoxin-induced model of Parkinson's disease in the top panel, significantly higher step-to-step variability in Parkinson's disease and Huntington's disease compared to saline-treated normal animals. Okay, so a lot of, I guess, standard parameters for DigiGait, and a nice set of growing publications looking at stride length, stride frequency, and so forth. We are doing more work with DigiGait to expand its scope and applicability for looking at, for example, how do animals encounter and negotiate obstacles. So we can't really introduce a banana into the path of the animal, but what we see here is the same mouse walking on the standard treadmill belt, and down below, after oil, mineral oil was added to the treadmill belt to essentially reduce the coefficient of friction and make for a slick surface for the animal. And I guess as one might expect, is we can see here the significant reduction in the stance width. So as the animal engages in the slippery surface, in this normal animal, as it engages the slippery surface, it has to change its posture in order not to fall. So changing the properties of the walking surface also primarily affected the forelimbs. And again, I suppose, given what we know about the function of the forelimbs, it shouldn't come as a surprise that the feedback mechanisms for the forelimbs were significantly reduced after walking on the slippery surface compared to baseline. So we see a good 30 millisecond reduction in the braking duration of the forelimbs, and hardly any change at all to the values for the propulsion duration on the slick surface. And what about obstacles? Well, there's all of us every day are encountering and negotiating things in our path. So we're also looking at how do animals negotiate obstacles. In this instance, we have the same mouse looking at two different conditions. One condition, we've introduced a simple piece of white tape on the treadmill belt, flush with the surface. And here, that same piece of tape with a slight lip. And when we watch the animation of the animal, when it negotiates or encounters the strip without a lip, it doesn't change its gait at all. It continues to power forward. In contrast, that same animal, when a ½ centimeter lip was added to the obstacle, we can see that the animal senses, encounters, and then changes its posture and its kinematics in order to negotiate that little jump. And then the same experiment, but in this case we had the animal at baseline, and then after an injection of a very small amount of ethanol. And we can see down here when the animal, the ethanol-treated animal encounters the jump, it doesn't have that same degree of pause and adjustment as baseline. There's a growing interest in developmental aspects of gait. So DigiGait is very useful in mice when they're about 2 weeks of age. Here we see significant postural and kinematic differences in mice at 2 weeks of age versus post-weaning at day 25. And this is very useful for looking at mouse models of cerebral palsy, muscular dystrophy, spinal muscular atrophy, just to name a few. DigiGait is now the most widely published treadmill gait analysis system available, including publications in Huntington's disease, Parkinson's, ALS, MS, OA, RA, pain, stroke, spinal cord injury, here the Rett syndrome mouse models, arthritis, TBI. And it's got, hopefully I've been able to describe to you its features. It's obviously in vivo, it's easy to use, it's very turnkey. Our success is based on your success, so we plug it in and make sure that you're generating data from it. It integrates several metrics that we've been measuring for years with other tools, and applications are many.
And today what I'd like to do is just briefly introduce you to amyotrophic lateral sclerosis and discuss the mouse models, or at least one mouse model that's used to study this disease in the laboratory, and then to go on and hopefully convince you of how important it is to characterize these models and to correlate what we find in with pathological changes with behavioral changes. And before I go any further, I feel I must acknowledge the folks who are actually involved in doing the work and our funding agencies shown here. In particular, I'd like to acknowledge Ron Oppenheim, my longtime collaborator who recently retired, Jim Karras, who's director of our ALS clinic, and of course Tom Hampton, who helped us with the behavioral analyses. So amyotrophic lateral sclerosis, ALS, or also known as Lou Gehrig's disease in the U.S., it's known worldwide as motor neuron disease, strikes about 350,000 individuals worldwide. We have about 30,000 individuals living with ALS in the U.S., with about 5,000 new cases each year. Here at Wake Forest, we see approximately 150 patients a year. The average age of onset for the disease is about 55, but as you can see from a patient that I show here, you can have patients as young as about 28 and as old as 80. Patients present with difficulties with speech and swallowing, muscle weakness, muscle fasciculations or twitching, spasticity. All of these symptoms correlate with a dysfunction and degeneration of motor neurons and eventually progress to where a patient is essentially paralyzed. While they're paralyzed, however, intellect, cardiovascular, and other bodily functions are not affected. The disease, with no substantially effective treatment, patients die on average about 3 years after diagnosis. And I think our patient here shows the progression of the disease. She got pregnant around the time that she was diagnosed, and you can see how her disease progressed as her child grew to be about 1 year old. About 90% of the cases of ALS are known as sporadic. That is, there's no family history, no definitive genetic association with the disease, although some genes that I have listed here have been associated with increasing chances of developing ALS. The remaining 10% of cases are familial, suggesting there's a genetic association with the disease, and several causes of familial ALS have been identified that I show here, with the most recent being the hexanucleotide repeat found in an open reading frame on chromosome 9. The first genetic association with ALS was found and reported in 1993. This was an autosomal dominant missense point mutation in SOD1. SOD1 is involved in the conversion, in regulating free radicals. It was found in about 10 to 15% of the familial cases examined. That translates to only about 1 to 2% of all ALS cases. But the mutation suggested a role for free radical damage in ALS. With that report in 1993, a year later in 94, we had the first reports of mouse models of ALS. And the one I will focus on today is the G93A mouse model. This mouse model is commercially available from Jackson Lab. And the animals developed symptom onset at about day 90. And what we mean by symptom onset is the inability of the mutant mouse to fully extend its hind limbs when it's suspended by the tail. These animals die between about day 120, 140. And the G93A mouse has been used in many preclinical studies. Here, as an example, I'm showing one of our publications where we treated the SOD1 mice with recombinant human heat shock protein 70. And what we found was treatment delayed symptom onset as determined by leg extension. Here's treated versus untreated animals. And treatment also extended the ability of the animals to run on the rotarod, as shown here by the dark line shifting to the right. It also showed an extension of survival in these animals. The muscle weakness that is the patients present with correlates to a loss of innervation of skeletal muscles, and the same thing happens in the G93A mouse. So by day 90, the medial gastroc muscle has lost about 60% of its innervation. Here's our SOD1 versus our wild-type animals, and we determine that anatomically by staining muscle tissue with a fluoroform labeled alpha-bungratoxin that will bind to the postsynaptic acetylcholine receptors present on muscle, and antibodies to vesicular acetylcholine transferase that will be present in the presynaptic terminals of the motor neurons, and neurofilament that will identify the axons. So when you have overlap of red and green shown here, we have an innervated neuromuscular junction. Lack of green, as shown in the lower panels, indicates a denervated neuromuscular junction. With human subjects or human patients, we also see a reduction in the number of motor neurons at end stage, and the same thing occurs in our mouse model. So at end stage, we can see about a 50% reduction in the number of motor neurons compared to the wild-type animal. There have been many reports, and I show just a very small sample here of what we could consider landmark papers in ALS. In fact, the first published report by Charcot was in 1874 describing ALS. And yet, in spite of this, and in spite of the availability of our mouse models, we still don't know when and where ALS pathology begins and what cells and processes are involved. So if we look at our timeline for the mouse model, we call up until from birth to P90 pre-symptomatic, and then P120 and on is considered end stage. End stage for humane reasons is when the animal is placed on its side and unable to right itself within 90 seconds. If you go through the literature, however, and look at what are the changes that have been reported in these animals, there's many changes that are reported earlier than P90, and that made us start to think, well, perhaps pre-symptomatic should be considered earlier. In fact, perhaps even up to the first postnatal month. We have some evidence for that, and here I'm again using one of our papers as an example, but when we treated the mice with recombinant human heat shock protein 70, we saw that we had the best survival of the animals when we treated earliest at P30. In another study done by Tom Gould in Ron Oppenheim's lab, what Tom did was that we know that the proapoptotic protein BAX is critical for motor neuron cell death. So if you count the number of motor neurons in a BAX knockout mouse compared to a wild-type mouse, you see that the motor neurons that would normally die during development do not die. So the BAX knockout mice have about 50% more motor neurons than wild-type. In SOD1, you have lost about 50% of the motor neurons. But if you cross the BAX knockout with the SOD1 mouse, what we found is that there's an enhanced number of motor neurons, and they did not die in these animals. And while the animals did have a slight extension of survival, while the motor neurons did not die, the mice died. And what Tom started to do was to see, well, what's going on in these animals? And what he found was loss of innervation of the medial gastroc muscle occurred and paralleled the wild-type animals in the BAX knockout SOD1 animals. And interestingly, what he observed was as early as day 25, postnatal day 25, there was already a statistically significant loss of innervation of the medial gastroc muscle. And so that got Ron and I to start thinking about, well, when and where is the earliest pathology? Motor neurons are complex cells. They have large cell bodies, extensive dendritic trees that sit in the spinal cord, and yet their axons and their terminals are located in the peripheral nervous system interacting with muscle. So we decided to try to begin a study to identify early changes, and we focused on two motor pools, motor neurons innervating the tibialis anterior muscle, and these were identified by retrogradely labeling the motor neurons with an injection of a fluoroform-labeled toxin into the TA muscle, or the soleus motor pool by labeling another fluoroform-labeled toxin into the soleus muscle. And you can see we can identify the two distinct motor pools. Why did we choose TA and soleus? Well, in the mouse, the TA muscle is composed primarily of type 2B and type 2A fibers, fast fibers, and we know in ALS that motor neurons that innervate the type 2B fibers are most susceptible. In the soleus, in the mouse, they're composed of type 1 fibers as well as some 2A fibers. And motor neurons that innervate the type 1 fibers are less susceptible in ALS. So the first thing we did was to look to see when does denervation begin in these two muscles. And as you can see here, percent denervation, day 14, day 30, and then closer out in toward end stage at day 100. And between 14 and 30 in the tibialis anterior muscle, by day 30 we have about 35% of the neuromuscular junctions have been denervated. Interestingly, in the soleus muscle, even out by day 100, there's very little denervation of neuromuscular junctions. All right, that's in the periphery at the muscle. What's going on in the spinal cord at the motor neuron? So we looked at the ultrastructural morphology of the cells. And one thing that really struck us, if you focus on these lower two panels, this is wild type, and the arrows are pointing to mitochondria, normal mitochondrial morphology shown here. But in the SOD1 animal, we saw these enlarged mitochondria. Sometimes they were showing slight vacuolization. If we looked at the distal dendrites of motor neurons, these are located in the white matter surrounding the lateral motor column. Wild-type dendrite shown here in the lower left panel. Here's a mitochondria. We look at the mitochondria in the SOD1 animal in the distal dendrites. You see they're showing gross morphological changes in greatly enlarged, very vacuolated mitochondria. We quantified this and found that at day 30 there are fewer mitochondria in the SOD1 animal compared to wild type, but while there were fewer, the sizes of the mitochondria were increased, as shown here with increasing numbers as the mitochondria area enlarged. Does that correlate to function of mitochondria? Well, we collected the ventral spinal cords, isolated mitochondria, and tested them for membrane potential and saw a decrease in the membrane potential in the SOD1 animals compared to wild type. We also had reduced ATP content in SOD1 and ATP generation in the SOD1 animals compared to wild type. We cannot exactly say that this is due to the mitochondria and the motor neurons, but it was just the motor neurons and their presynaptic terminals, or the presynaptic terminals of the inner neurons, that only showed the mitochondrial changes. No other cells showed mitochondrial changes. So we think this decrease in function correlates to those pathological changes. Does it occur just in the spinal cord, in the dendrites and the cell body? Well, no. We looked at axons of both the TA innervating motor neurons and soleus innervating motor neurons, and you can see that their mitochondria also are showing these pathological changes. At the presynaptic terminal, this is a presynaptic terminal, the motor neuron here, postsynaptic muscle here, and the mitochondria in these presynaptic terminals also show pathological changes. And in fact, when we counted mitochondria in presynaptic terminals of both soleus and TA, the numbers were decreased in SOD1 animals, but the sizes, the general area of those mitochondria was increased, both in the soleus and the TA presynaptic terminals. Was it just mitochondrial changes that we saw? Well, we also looked at synapses. So here we're just looking at ultrastructural characterization of synapses, and what we did was count those on the motor neurons. So this is from a wild type animal. Here's the synaptic density, presynaptic terminal in this area, postsynaptic on this side. We know it's a C-type synapse because at the synaptic density, you see at the postsynaptic terminal, the rough ER abuts right up against that synapse. These are generally thought to be excitatory synapses. In panel B, we have the presynaptic terminal, motor neuron cell body here, synaptic density. The vesicles here are varied in shape and sizes. They're known as pleiotrophic or type 2 synapses. These are generally inhibitory. In panel C, I show presynaptic terminal, postsynaptic region. Here's the synaptic density, and the majority of these vesicles are round. These are known as type 1 or excitatory synapses. So counting those on the motor neuron cell body, the axosomatic synapses, we see that there's really no change in the number of total synapses, but there's a decrease in the type 1 synapses, no change in type 2, and an increase in the C-type terminals. In the axodendritic synapses, there's an overall decrease, and I'm sorry I lost my asterisks here, but there's an overall decrease, significant decrease in the total number of synapses. And while there's no change in the type 2 or the inhibitory, there's actually a decrease in the type 1 or excitatory synapse. These are at day 30. So by postnatal day 30, there are obvious pathological changes both in the dendrites, the cell body, axons, and terminals of motor neurons. There's denervation, but only in the tibialis anterior, not in the soleus, even though those pathological changes that I mentioned were observed in both TA and soleus motor neurons. And these early changes got us to really think, well, what does this mean? Is this significant? And how could it be significant if symptom onset is actually not beginning until day 90? Here we show the decrease in the ability to run on the rotarod occurs in the 90s. And then again, the failure to fully extend the legs is detected around day 90. Well, that made us start to think, how does this correlate to our patients? So in the mouse, if it cannot extend its legs, you would expect that a patient in a similar situation would have great difficulty in walking. And we thought and looked at what happens to patients. And here, as an example, I show John Blaze. John Blaze, or better known as the Blazeman, was diagnosed with ALS, and he was an endurance athlete and determined to run the Ironman, which in fact he did after being diagnosed with ALS. You can learn more about John at this website. But what John found when he was first diagnosed was that he was having difficulty holding things such as soap or a toothbrush. If you look at ALS's website, it also notes that the initial symptoms of ALS are varied between individuals, but they're generally very subtle. In fact, Jim Karras, the director of our clinic, tells me that patients come in with difficulty turning the key in the car or tripping because of slight foot drop. These are not profound inability to extend hind limbs, but more subtle behavioral changes. And that's what we began to ask. I don't know about others in the audience, but I like to say it was sort of the duh moment for us when we thought, well, you know, our mice have not been telling us, hey, folks, investigators, I'm having a little problem walking in the cage here. You know, my foot, I seem to be tripping a little bit. We just don't observe these changes when you're looking at the mice. So we wanted to look at a more sensitive behavioral analysis, and Gait is the obvious choice. And that's when we met up with Tom Hampton, who introduced us to DigiGait. And Tom went through the many aspects and measurements that you can do at DigiGait, and I just want to focus on two that we focused on, and that was hind limb stance and paw placement angle, and thinking that these two motor functions could be affected if there's denervation in the tibialis anterior muscle. And what we found was, yes, hind limb stance width changes in that can be detected early. In fact, the first detectable difference was observed at 32 days. And then when you looked at variability in paw placement angle, perhaps a little bit of foot drop in the mouse, you see that that variability increases at day 28 and day 32. So these behavioral changes that we're observing did in fact correlate with some of the pathological changes that we see. And I think what I hope I've gotten you to think about is that in the mouse model, the pathological changes are probably occurring much earlier than the overt behavioral changes, such as failure to extend the legs, that it's very important to thoroughly characterize the pathology in these models and to utilize the appropriate behavioral analysis to correlate with those pathological changes. And I really believe that this is what we're going to have to do if we're going to understand the earliest pathology, which is what we're going to need to gain insight into potential causes of the disease, to identify effective biomarkers, and ultimately to develop effective therapeutics. And thank you. I want to thank you for listening. This was informative.
So most toxin models are use a more acute or subacute methodology. And this results in quite a significant loss of dopamine, either unilateral or bilateral. And again, it's relatively easy to see motor dysfunction in these more acute models. But we've developed a more progressive model to try to simulate in the Parkinson's patients the longer-term progression of the disease in terms of dopamine loss. We don't get quite as much dopamine loss as in the acute models. We get somewhere between 60 and 80% loss. And I think what we're finding is that the slower loss is resulting in the brain adapting to these changes. And therefore, it's been a challenge, to put it mildly, in terms of looking at motor dysfunction, in terms of what to measure. And the other thing which I'll be presenting to the audience is some of our treatment strategies in terms of looking at treatment, whether it's drug treatment or exercise. We've taken an approach where we're looking at intervention or restoration versus protection. A lot of studies that are published will treat the animals first with whatever compound they're interested in and looking at neuroprotection, but we have this bias that it's not clinically relevant. And therefore, reviewers, to be honest with you, reviewers have been challenging me to look at not just intervention, but mainly restoration. And again, I'll describe some of those studies in a moment. So first, the model. So Natalie Goldberg was the lead on this study that we published back in 2011. So again, comments from reviewers asking, we want a more progressive model of the disease, and one did not really exist at the time. And so we've taken the approach of looking at MPTP, looking at higher doses of the drug over a weekly basis. The initial study looked at 4, 8, 16, and 20, and 32 milligrams per kilogram per week, 5 days per week. Because we've actually changed the source of the MPTP, where we purchase it, we're now looking at slightly higher doses. We're actually looking at 10, 20, 24, and 32. 32 is still the highest dose that we use, and that's pretty much what the animals can tolerate. But as you can see here, both looking at tyrosine hydroxylase immunolabeling here in the substantia nigra, or tyrosine hydroxylase labeling in the striatum, you see a progressive loss of dopamine, of TH, within the striatum, or within, as you see here in the upper panel B, the loss of dopamine TH-containing cells over this time period. And of course, we're biased in terms of the fact that we think one of the beauties of this model is that you can intervene at any point during the progression of the loss of the dopamine in terms of some sort of treatment. So what we've started doing is looking at either intervention halfway through, or I'll present data a little bit later, looking at actual restoration, where we actually give all 4 weeks of MPTP followed by drug treatment. So this was the initial study. Also at that same time, I met Tom actually at a meeting in Chicago where this was the first time I had seen the Digigate machine, and we hooked up with him to actually collaborate on a study. Again, Natalie was the lead on this study, published several years ago, in which we looked at gait function in these MPTP treated animals. So we gave the 4 weeks of MPTP, then we shipped the animals to Tom, and I went out there to observe what this whole Digigate machine was all about. And so the animals were tested flat, essentially, and I'll bring that point up a little bit later because we've changed our methodologies a little bit. And as you can see here from this publication, that we saw several measures that were changed following the last dose of MPTP. Stride length, frequency, total number of steps, duration, and so on. So we were very excited that we could, that using this machine, that we could pick out these very small changes. And as Tom said in his introduction, as in patients, you only sometimes see a 10% difference. And so, and again, our standard errors were incredibly small. So we were just really duly impressed with what this machine could do. The other important thing is that it took away any sort of operator issues. You essentially just put the animal on the machine, you turn the machine on, you record the steps, and you do the analysis. So as opposed to many other behavioral tests that are more operator dependent, sorry about that, this took away that potential problem, because as we know, each assistant or graduate student, they have a different feel for the animal. So I wanted to be able to take that away as a variable. Okay, so this was our first publication with Tom back in 2011. Subsequent to that, we've looked at the effects of various treatments in our model. So this was an intervention treatment with a compound that we've become very interested in called 7,8-dihydroxyflavin. Again, Meshul Sconce was the lead on this study that was published this past year. So in this particular study, there were only a couple of measures that were actually statistically different, which is another thing I want to point out, is that in this particular study, our initial study, in looking at the amount of loss of tyrosine hydroxylase, we got somewhere between 60 and 75% loss of tyrosine hydroxylase. So just keep that in the back of your mind. In this particular intervention study, we got about, again, depending on whether we do immunohistochemistry or Western blot analysis, somewhere between 60 and 80% loss of tyrosine hydroxylase and about a 55% loss of TH cells within the substantia nigra. So that's just something to keep in the back of your mind. We are getting variable amounts of loss, and this was an issue. The other thing I want to point out compared to this study, in this particular study, we had the Digigate elevated up in the back. So the animals were actually running in a decline position. It's elevated about 10 inches, something to that degree, and so the animals are actually running downhill. So in this particular case, following MPTP treatment, we saw an increase in the hind dual stance time, and that with dihydroxyflavin treatment, that decreased back to essentially the vehicle level, as you can see here in A. In B, this was an interesting finding, again, something that we hadn't seen before, is looking at whether the forepaws or the hind paws, the distance away from the midline, the difference between, again, the hind paws and the forepaws and the hind paws. And what you can see here, that in this particular study, after 4 weeks of MPTP treatment, that there was a significant increase in the distance from the midline of the forepaws compared to the hind paws. And that's noted here by the negative number. And again, with the drug treatment, that was essentially reversed. One can do all sorts of correlations, and again, reviewers love to see these kinds of correlations, and so do we. And so we looked at several biomarkers, including tyrosine hydroxylase. We looked at a growth factor called superior cervical ganglion 10, or SCG10, in the substantia nigra. And we're also interested in the second messenger system. And so we looked at the active form of TrkB, which is the receptor for brain-derived neurotrophic factor, or BDNF. And again, you could see these very nice correlations between the various gait measures and the biomarkers that we were looking at. Again, this is published, so the audience can, if they're interested, they can find more details in terms of the paper. Another interest of ours is looking at the effects of exercise. Again, this was an intervention study, so the intervention study is 2 weeks of MPTP, then we start the exercise study. And in this particular case, it was voluntary exercise on a running wheel. And again, Michelle Sconce was the lead on this particular study that was published earlier this year. And so in this particular study, in which I'm looking at my notes here, we found about a 60% loss of dopamine cells and about a 70% loss of dopamine terminals in the striatum. Again, the measures that you see here are different than what we found in the previous study here. So again, each study is producing slightly different changes in gait measures. In this particular study, Michelle found changes in paw area, stance width, shared stance, and another interesting gait measure is maximal rate of change of paw area in contact with the belt during the braking phase of their movement. And again, following the 4 weeks of MPTP treatment, as you can see here in pretty much all of the measures, there was either a significant increase or in terms of the shared stance, there was a significant decrease. And the good news is that with this dihydroxyflavonoid treatment, it reversed back essentially to the vehicle levels. And I'm sorry, not dihydroxyflavin, I'm sorry, exercise. Thank you. Yeah, so this was exercise. And again, just similar to the dihydroxyflavin, exercise was also very effective. And again, looking at several correlations with the various gait measures that were changed, we're very interested in looking at glutamate measures within either the substantia nigra or the striatum. In this particular case, it was the substantia nigra. And again, looking at the vesicular glutamate transporter 1 or another one called GLAS, the glutamate aspartate transporter, we saw a very nice correlation between levels of these biomarkers and improvement in motor function. Another study that we did in collaboration with a group at Oregon Health Sciences, the lab of Vivek Uni, Katerina Spinelli, who was the postdoctoral fellow who was the lead on the study, and using another model of Parkinsonism, which is their alpha-synuclein transgenic mouse, which overexpresses alpha-synuclein within primarily the cortex. Sorry about that. What Katerina found was with curcumin treatment that there was improvement in several measures of gait, as you can see here. And again, this paper was recently published this year also. In a more recent study, again, Michelle Sconce was the lead on this study, we looked at the effect of restoration. So again, 4 weeks of MPTP treatment followed by 4 weeks of this flavin administration. And what Michelle found was after MPTP treatment, as you can see in this column here, there were several measures of gait that were statistically different. And in this particular study, I'm looking at my notes here, she got even a greater hit in terms of dopamine loss. So somewhere between almost 80% loss of dopamine in the striatum and about a 60 to 65% loss of dopamine cells. So again, a greater hit in terms of dopamine loss, and as you can see here, there were several more measures that were statistically different. And again, following the flavonoid treatment, pretty much all of them reversed back to the level found in the vehicle group. So again, not only with an intervention study did we see a blockade of further loss in several measures, but I think the more important study is restoration, that actually after the loss we can reverse some of these changes. And again, looking at several measures, looking at several correlations between several of these gait measures, and I'm just showing a few of them here, and several of the biomarkers we're interested in, again, TRAC-B, AKT, ERK, SEG10, we see very nice correlations between the behavior and the levels of these biomarkers, which again all improved following dihydroxyflavin treatment. In a a recent study that actually was just submitted for publication this morning. So not only is it just, it's not even a manuscript in preparation anymore, it was just submitted for publication. Madeline Churchill in my lab was the lead on this particular part of the collaborative study with a colleague in Italy. And we're looking at the effects of cyclosporin following MPTP treatment. So again, it's a restoration-related study. And what she found here, now in this particular study, the hit wasn't as big in terms of the dopamine loss. We found about a 65% loss of dopamine within the striatum, but only about a 45% loss of the dopamine cells. And so in this particular case, there were two measures, stance width and step angle variance, that were statistically increased following MPTP treatment and then reversed following cyclosporin treatment, as you can see here. And again, as Tom pointed out in his introduction, you can see how small the standard errors are. It is really amazing that with a behavioral study that the variability is really, really small in these animals. And in discussion earlier, actually last week with Madeline, in going over all of the data, one of the things that she came up with was the idea that many of these measures relate to gait instability. And so with MPTP, you see a lot of gait instability that's corrected following various drug treatment or exercise. And so this is just to give you kind of an idea as to the kinds of studies that we do in the lab in terms of our Parkinson's disease model, and actually the usefulness of the Gait, the DigiGait analysis. And so I think in conclusion we can say that at least in our MPTP model, most of the measures that we see changing, and again, the unfortunate thing is that you get different measures changing depending on the experiment, probably dependent on the loss of dopamine, as I point out here, is that all of these measures relate to gait instability. And perhaps the variability that we see from study to study is related to the degree of loss. And again, even though we're using the same type of animal, about the same age, the same dosing, the same sex, they're all males, by the way, we do see variability. And you know, kind of the way we're looking at this is the fact that even in patients with Parkinson's disease, there's variability from patient to patient. And that, again, in our model, we're very much looking at restoration, so we give the MPTP for 4 weeks, and then we treat the animal for an additional 4 weeks. So, again, we're looking at real restoration in the sense of the patient coming into the physician, being diagnosed, and then what do you do? So this is the model that we are now using, and that the variability that we see in the Gait analysis probably has something to do, as we see in patients with Parkinson's disease, that they also show variability also. So again, thank you very much for your time, and be happy to take any questions.
All right, fantastic. Thank you so much, Charlie, for that great presentation. That was very, very interesting data, and we are, running a little bit late. So what I'm going to do is we're going to jump right into our Q&A session today. I'm unmuting our other two speakers, Carol and Tom. So welcome back to both of you. We've had a lot of great questions. Charlie, just to sort of verify what you just talked about. You talk about each study producing different changes and some variability. You indicate a lot of, one of the challenges you face, I guess, with gait analysis is that oftentimes subtle changes in gait are unpredictable. And right. So can you just confirm then that this is sort of, they mirror maybe some of the observations that you see with the clinical condition as well?
Well, not being a physician, when I go to my journal clubs, I think physicians are always talking about, especially some of the data that they show, they always, show a wide range of gait changes in these patients. And so I think the rationalization with our study is that we're seeing these same gait changes, perhaps dependent on the degree of loss of the dopamine. And I must admit that this is something that we didn't really appreciate until more recently, until we started actually looking at our data to see what to what degree do we see loss? And like in one of the studies I mentioned that Meshul carried out, that we're in the process of still writing up, is that she got a very big hit of about 80% loss. And as you could see in one of the in one of the tables, there were like 12 different gait measures that were significantly different. And, you know, so in fact, I was just talking to Madeline this morning about doing a correlation between the degree of loss and the gait changes. So I think that's something that we're in the process of thinking about doing.
Okay, perfect. Carol, do you have any comments on sort of, again, the subtle changes that you see relating to the clinical condition that you're studying?
No, I think looking for these subtle changes correlates best, as Charlie went through, correlates with the variability and the subtle changes we see in patients. And the other thing is, the pathological changes that we examined, it's very timely to look at those, and takes a lot of manpower, hours, and obviously funds. But if you want to do treatment, the DigiGait is great, because as we treat, we can monitor our mice continually throughout treatment and see how they compare with gait changes versus untreated animals. So, there's a lot of benefits to focusing on the subtle behavioral changes and seeing if treatment can affect that.
Okay, great. Tom, this is sort of a culmination of a few different questions, and then also maybe some of my own thoughts. You've mentioned in your presentation that with the DigiGait system, it's possible to change speeds of the treadmill, increase or decrease the angle of the belt, and Charlie alluded to this as well, so that animals can either walk or run up an incline or down a decline, and also you can add obstacles to a protocol so that the animal can avoid those obstacles. So, I mean, altogether, why are these features so important for the study of gait, in your opinion?
The ALS model, Carol's model, the SOD1 G93A, that's a great example. When we first studied that model years ago, right around the time that we developed DigiGait, the early phenotype at a comfortable walking speed with animals walking horizontally, we actually found, and this was since published, that those animals were what we called supranormal. A very small window of time before the animals became quite sick, their gait was quite robust. And that was a paradox that, until we studied the pathology and read up on the literature, it made sense. Motor end plates get bigger, there's hyperexcitability of motor neurons. To see that paradoxical supranormal gait just before they capitulate to the disease phenotype was confusing, but it also taught us something. It taught us about a manifestation of ALS, and that's potentially one reason why it's gone missed in humans pre-symptomatically, and dovetails somewhat with what's known about the etiology of the disease. There's a high incidence of it amongst athletes. So the second time we did the study with now Carol's mice and Ron Oppenheim from Wake Forest, armed with the knowledge of that paradoxical supernormal gait, we challenged the animals. Knowing that they're going to do quite well through 13, 14 weeks of age, we instead challenged the animals to walk up an incline at a fast speed. And it was there that we saw those, as Carol showed, the changes in the narrowing of the stance width and the changing of the paw placement angle. So only when we challenged the animals were we able to see the defect, and quite early.